It's a pleasure to introduce a fellow biotech veteran who probably has almost as many scars as I do from 20-odd years in this field. Anne was appointed the Chief Operating Officer of Stem Cells, Inc. In November 2006. Prior to that, she served as Vice President for R&D, and she has more than 20 years' experience in the field of stem cell biology. In fact, was the co-discoverer of human hematopoietic stem cells when she was at Systemics, a pioneering company really in the cell therapy business in this area. Under her direction at Stem Cells, Inc., the scientific team has discovered the human central nervous system stem cell you just heard about. They've also discovered a population of human liver and grafting cells and a candidate pancreas stem cell. Dr. Tsukamoto received her Ph.D. from the University of California, Los Angeles, and did postdoctoral research with Harold Varmus, who was then at UCSF. In Harold's lab, she worked on the Wnt1 gene, which is a key player in stem cell self-renewal pathway. She's an inventor of half a dozen U.S. patents related to humans' hematopoietic stem cells, and it's wonderful to have you here and learn about this latest chapter in your illustrious career. Thank you for joining us today. Anne?
Thank you very much for this opportunity to be here. It is truly a wonderful chance to sort of just give you the very highlights of the work we've done on the human central nervous system stem cells. So you've heard from Stephen that we are in clinical trials, and I just want to give you an overview of what it took to get to that point in time. So I'm going to talk about the isolation and characterization of the human central nervous system stem cell. So we started with a technology platform that was actually developed here at Stanford by Irv Weissman in his lab when they had identified the mouse hematopoietic stem cell. So we modeled our discovery platform after what was developed in his lab. And that is to use monoclonal antibodies to surface markers and identify a rarer and rarer population of cells by cell sorting, and to test these cells in various in vitro and in vivo assays. So our starting point is the mature tissue, and we make a single cell suspension. We have both proprietary and known monoclonal antibodies that mark antigens on the surface of the cells, and we use the high-speed cell sorter in a reiterative process till we get to a rarer and rarer composition of cells that in both our in vitro and in vivo assays define a population of stem cells. So as Ed mentioned, we've done that for the human central nervous system stem cell. We have a candidate liver engrafting cell, which we hope to talk about one day, and a candidate pancreas stem cell. But today's focus is on the human neural stem cell. We've shown that at the single cell level, that these cells, when put into culture, can expand in a process called self-renewal. And when you take those neurospheres that have developed from the single cell and you put them under differentiation conditions, they will differentiate into the neurons and glial cells, which are the main cell types of the central nervous system. So we've elevated these cells to the stem cell category because we've shown at the single cell level they're capable of self-renewing and multilineage differentiation. So that's in culture. We use this methodology to then develop cell banks for the clinical transplants. So it's a two-step process to develop the cell banks. The first step is the purification by cell sorting, and you see in the very left panel the purified populations of neural stem cells. The second method is to expand these cells specifically in culture, and here in the middle panel you see some of the neurospheres that are growing in culture. This two-step process allows us to reproducibly generate these banks of human neural stem cells. And that's very important because if we were to just take the brain tissue, make cell suspension, and put them in culture, we don't get the reproducibility of generating the banks that would be required for a large-scale manufacturing process. And you can see that starting with several different tissues, you see the reproducibility of the growth of those cells in culture. We've shown that these cells, when grown as neurospheres, maintain a normal karyotype, don't form tumors in vivo, and they retain their multilineage potential. And they do not require pre-differentiation before transplant. So there are many steps to get from the research bench to a GMP cell product that you're going to transplant into the patient. But I'm just going to highlight some of the very key aspects to get past the FDA regulations. We need qualified raw materials, and one of the most important is the source of the donor tissue. There are new good tissue practice regulations requiring all the donor consents. So the qualified raw materials include the donor tissue, the growth factors and antibodies that you need to make. So you have to make each of those under the defined regulations. The process has to be adequate to cover whatever you plan to use in the clinic, and it has to be done under containment in a sterile environment in what they call the good manufacturing practice conditions. You have to perform an extensive series of release testing so that you know that none of your cells could potentially harm the patient. These include a series of microbiological tests as well as virology tests, and that release testing takes 4 to 6 months for every new cell bank that you make. And then because this isn't a renewable, it's a renewable source, but it's not forever. It's not an infinite source, so you're going to have to make new cell banks, and you're going to have to demonstrate that each lot of the cells performs comparably. And so you have to go through an extensive series of, again, in vitro and in vivo tests to show comparability, and those tests can take up to 9 months. So just to highlight that because of this process, we've shown that there is lot-to-lot comparability. We look after the expansion of these cells, and we show that those cells maintain the stem cell marker, CD133 and nestin, so very high purity of those cells. They don't express the hematopoietic marker, CD45, and they haven't differentiated down the neuronal lineage. They maintain their stemness in culture. But then upon differentiation, we show that the cells which are that don't express the neuronal markers before you differentiate, reproducibly we show that they can differentiate down the neuronal lineage. So what happens when you transplant these cells into the immunodeficient animal model that you need to show engraftment? We have to use the immunodeficient mouse model because it's a xenogenic transplant between human cells into a mouse model. And if you don't put them into this immunodeficient animal model, you will get a process called xenorejection. So we have transplanted these cells, and you can see from all of the brown staining in this picture, this is a sagittal cross-section of the mouse brain, all of that brown staining are the human cells that have arisen from a single transplant of the human neurostem cells at birth. The cells, whoops, oh, I must have done something, sorry. The cells seed the endogenous mouse neurogenic niche, and they proliferate slowly, much as you would expect for what the mouse stem cells do within the mouse brain. The human cells also migrate along the same rostral migratory stream that the mouse stem cells would take, and then they go to the olfactory bulb, and there they differentiate into mature neurons. So we've looked at various parts of the brain, and in a very site-specific manner, we see that these human cells have engrafted into the mouse brain, and they take the signals from the environment of where they land, and they become the right cell types in the right zip code, as we say. So they form neurons in the olfactory system, astrocytes near the subventricular area, and they form the myelin-producing oligodendrocytes in the white matter areas of the brain. So how did we go about choosing a disease or injury target? When we first knew the characteristics of the cells, we held a series of meetings with scientific and medical experts, and there were over 50 different disease targets that we looked at that affected the brain or spinal cord. And it was a very long process because we had to include animal models in assays, and we had to know that those animal models really recapitulated the human disease. So our objectives were to be able to demonstrate a mechanism of action for these cells in preclinical animal models in, of course, a very aggressive time frame since we were a small biotech company. And we also had to know that it was feasible to treat that patient population. So after a series of meetings, we landed on a neuroprotection strategy through the delivery of soluble proteins, and we considered that the lowest hanging fruit. And the top of the list was the NCL, or the Batten's lysosomal storage disease, and you've heard from Stephen some of the rationale for that. As you know, it's always fatal. There are known genetic mutations which can be tested for, and there was an enzyme assay that we could use to demonstrate that our cells produced this enzyme. The NCL animal model that was available, the PPT1 knockout mouse, really mimicked the human disease pathology, and that was very important to us. In the bottom corner, you see that our human cells express that PPT enzyme at significant levels. So we knew that that was the first hurdle. The in vivo studies, I showed you in the previous slide that we had very robust engraftment and that and the migration throughout the brain. So the concept that these cells could serve as cellular mini pumps in the brain was the first demonstration to show that the cells could engraft and migrate throughout the brain. And the other point is that many people have tried to transplant human tissues in the Parkinson's model, and it's very difficult to show neuronal integration. So we thought, let's choose a target that wouldn't require neuronal integration in the first try at bat. And it's a homologous use of the cells. So we have brain-derived cells going back into the brain, or central nervous system cells going back into the CNS. And it does then have the potential to provide the normal cells in a cell replacement strategy. We weren't trying to do these cells in a non-homologous use. So here is some of the in vitro data in culture, where we show that these human neural stem cells, when put into culture, could produce the enzyme that could be taken up by the mutant host, the mutant patient fibroblasts. So in green, you see the level of the enzyme in the mutant fibroblasts alone. The red shows you the uptake of the enzyme in a co-culture system where the cells don't come in contact with the cells. It really has to be secreted across the membrane and have to be taken up by the mutant cells. And if you add an excess of the receptor mannose-6-phosphate, you can block that uptake. So that showed that the mechanism was potential. So what happens when we transplanted them into the brain of these PPT1 knockout animals? We show here that in red that the transplant of these cells was able to reduce some of the storage material in the disease. You can see that compared to the gray. And when we looked at the ability to protect the host neurons from cell death, in the next slide, I'm going to show you a normal non-skid mouse brain compared to the knockout animal that hasn't been transplanted. And here you see that in the normal mouse brain, if you focus on this particular area of the brain called the hippocampus, you see that in the CA1 region, you can actually count the number of neurons in that area of the brain. And when you look at the PPT1 knockout animal, you see that there is a virtual absence of those cells. And so in the CA1 region, there are only 8% of the host neurons that survive in this animal model without a transplant. When we look at the animals that have been transplanted, first you see in the bottom that the non-transplanted animals have a very low level of enzyme, and when we transplant either a low or high dose of cells, you can increase the level of the enzyme, the missing enzyme, to 33 to almost 60% of normal. And when you look at the hippocampal area, here again there's the non-transplanted animal for comparison, and here are two sections from both low dose and high dose animals that have been transplanted. And you can see that there is a statistically significant difference in the number of cells that remain in this portion of the brain, where 57% of the cells are remaining after the transplant of these human neural stem cells. So from this data, we see that there is a neuroprotection of the host cells. This slide shows another area of, two areas of the brain, the CA2-3 region of the hippocampus and in the cortex. And you can see that in the CA2 region, you're up to 92 to 97% of normal. And in the cortex, at the high cell dose, we didn't have animals in this cohort. You see that you're protecting about 77% of the host cells following transplants. So that was the preclinical data that we used to submit our IND filing to the FDA. And what are some of the critical factors for translating into the clinics? We had rigorous in vivo characterization of the cells. We had made in research capacity more than 50 different cell lines. We've shown in greater than 2,000 animal mouse brains that these cells performed reproducibly and made the main cell types of the CNS. What was critical for us was the support from the NCL patient advocate groups, because they really bridged us to the academic researchers who had the animal models, the antibodies, and the enzyme assays, and the cell lines that we needed to perform these studies. And then, as I've just showed you, we had to have sufficient supportive data in a preclinical animal model for the FDA. We had to develop the cell banks and have them all release tested, and we had to have the capacity to scale up and produce the cells under the GMP regs. And we had to identify committed clinical sites and investigators. This slide shows you essentially a timeline. We published the identification and purification of the human neural stem cells in 2000. We did the characterization and testing in preclinical animal models from 2000, and this continues. It's ongoing because, as I mentioned, there were over 50 neurological targets, diseases and injuries that we've looked at, and we keep putting these cells into the various animal models. But when we put our stake in the ground in 2003 with Batten disease, we developed the protocol. In 2005, we sought regulatory review and we received approval. In 2006, we received approval from the OHSU IRB. We initiated the trial in 2006, and as you see, we'll complete the trial in 2009. So just as a brief summary, I've shown you data that it's just the tip of the iceberg, that our human neural stem cells produce the three main cell types of the central nervous system. We've applied it to a neuroprotection strategy in our first clinical trial in NCL. There are other lysosomal storage diseases that we could apply this to, as Stevens alluded to. We have announced that we have a collaboration with the KCI Institute for retinal protection using these cells. And so what are the other lysosomal storage disease that we can think about? This pie chart shows you the vast number of lysosomal storage disease. The ones in red have primary manifestation in the central nervous system. The two in yellow are the ones that we are targeting in our first clinical trial. We've tested our cells for the production of three other lysosomal storage disease enzymes, and we have shown that our cells produce those enzymes, and those are for Sanfilippo, Hurlers, and Gaucher's. So as you would expect, these neural stem cells, because the lysosomal enzymes are housekeeping enzymes, our cells produce them. So I'll just end with a last slide. We are targeting neuroprotection through another strategy, which is remyelination. If you were at the ISSCR meeting, we had presented data on, Eileen Anderson, our collaborator, has presented data on spinal cord injury. And we are targeting another rare and fatal disease called Spalitsius-Merzbacher. We have preclinical proof of principle that our cells robustly myelinate in the appropriate animal models. So thank you very much.